Good morning. Today is Friday, June 19th, 2020. My name is Ricardo Sandoval, and this is lab number 10. This lab is entitled Antibiotic Sensitivity, Kirby Bauer Diffusion. This is the experiment video for lab 10, and this is the final lab video for Biology 225 for microbiology for the summer 1 term at Los Angeles Pacific University. This is for week number 7. So here's a quick little sign I made, just summarizing everything I just said. Title of the lab, experiment video, my name, class, week, and today's date. So in this week's lab, what we're doing is we're playing or testing out antibiotics. And as we all know, antibiotics are what we get when we have a bacterial infection. So of course, there's a difference between bacteria and viruses. For example, when you get the flu or COVID, as we're dealing with now, both influenza and SARS-CoV-2, as COVID-19 is formally known, are both caused by viruses. While on the other hand, the microorganism or the microbe we'll be dealing with today, Staphylococcus epidermidis or S. epidermidis, is a bacteria. So when you get a bacterial infection, like let's say you're in the hospital and you have an IV in your hand that's been inserted by a catheter, or you have a chest tube, Staphylococcus epidermidis or S. epidermidis is normal flora, so it exists on our skin, but it can become pathogenic or infection-causing, malady-causing, I guess, when we have an impairment to our skin, such as an IV catheter, a central line, chest tube, mainline catheter, PICC line, all those kinds of things. So you're in the hospital, you have an IV, let's say, and it turns out that you start getting a fever or a cough, or you're just not feeling well, and they run some blood tests, and it turns out that your white blood cells are very high. They run a culture and sensitivity test, and you are infected with Staphylococcus epidermidis. Now that we know that it's a bacteria, your doctor or nurse practitioner, given whoever's taking care of you, or PA, will then prescribe an appropriate antibiotic to take care of you, to heal you. On the other hand, if you are infected with influenza, the flu, or SARS-CoV-2, COVID-19, antibiotics are not useful because antibiotics don't do anything against viruses, which is why we have bacteria, let's say, that have now become resistant to certain antibiotics. So some terms I want to talk about that I'll be mentioning here. So we have bactericidal and bacteriostatic. So bactericidal, the suffix "cidal" kind of sounds like suicide. So bactericidal means that an antibiotic will kill a particular bacteria, versus bacteriostatic, again the suffix static, or think stasis, anything that's bacteriostatic stays. So an antibiotic will just prevent further growth or further spread of a bacteria rather than killing it. So in this lab, this experiment, what I'm going to be doing is I'm going to use a petri dish, a big one this time, and using the microbe S. epidermidis or Staphylococcus epidermidis, I'm going to expose it to 3 different types of antibiotics: penicillin, gentamicin, and novobicin. Now, when it comes to antibiotics, we have 2 types. We have broad spectrum or narrow spectrum. And in a clinical setting, I'm a nurse, so broad spectrum, as it sounds, broad spectrum just kind of kills everything. So if you go into a hospital, for example, or a nursing home where I work, and you just start getting a fever, cough, diarrhea, all sorts of problems, and we yet don't know what's wrong with you as far as what bacteria is infecting you, or if you have a bacteria versus a virus. And the goal is to prevent you from getting sicker. Your doctor may prescribe what's called a broad spectrum antibiotic just to kind of kill whatever's there. Clindamycin, for example, is broad spectrum. Meropenem, Procefin, those are really common broad spectrum antibiotics. The 3 antibiotics that we'll be dealing with today are all narrow spectrum. So again, by the term narrow, they're very targeted to a certain type of bacteria. So when we talk about bacteria, we talk about gram-positive and gram-negative. And as we learned several weeks ago, the big difference between gram-positive and gram-negative is something called peptidoglycan. So when we're talking about a bacterial cell, there's part of the cell that's called peptidoglycan. Those bacteria, or those cells that are gram-positive when stained with that Fourier stain process, hold the color, hold that crystal violet thing that I was using, hold it really well because they have a very thick wall of peptidoglycan, versus something that's gram-negative that does not hold the stain well because their wall of peptidoglycan is very thin. So when treating a microbe in medicine in a clinical setting, we'll have better success treating something that's gram-positive versus gram-negative, because a gram-positive cell or a gram-positive microbe, just like it holds the stain better, it will take in more of an antibiotic, therefore killing or stopping the growth. So let's talk about the 3 antibiotics that I'll be using today in a bit more detail. So again, it's penicillin, gentamicin, and novobicin. So penicillin, we all know of, it's kind of like a home antibiotic, so to speak, in that there are many variants of penicillin. So penicillin, again, is what's considered a narrow spectrum antibiotic. And it's bactericidal. So like I said, bactericidal kills the bacteria. And it's mostly targets gram-positive bacteria. So again, bacteria that have a thick peptidoglycan cell. And then we have novobicin, that's bacteriostatic. Again, bacteriostatic just stops the growth of bacteria. It doesn't kill, so to speak. And it attacks what's called DNA gyrase. And DNA gyrase, again, the ASC suffix, lets us know that gyrase is an enzyme. And it's an enzyme that's necessary for DNA replication. So what novobicin does is, since it attacks that DNA gyrase, it stops DNA replication. You can't replicate DNA, the cell will eventually die and you will return to health. And then gentamicin, again, narrow spectrum, and it's bacteriostatic, just like novobicin, so it stops the growth, doesn't kill it, and it attacks gram-negative bacteria, so bacteria that has a thinner peptidoglycan cell, and it also attacks gram-positive staphylococcus. So when we talk about bacteria, there's the term taxonomy. That's, it's kind of how we classify things. So, I've talked about epidermidis a lot in this lab so far. So the first part of the S part of epidermidis is Staphylococcus. So Staphylococcus is the genus, or the most top portion, if you will, of the Staphylococcus genome, organization, whatever. And then we have epidermidis and other simple things. So we did learn in the gram staining portion of this that S. epidermidis, or Staphylococcus epidermidis, is gram positive. So knowing that gentamicin attacks gram positive Staphylococcus bacteria, we have a pretty good chance here of seeing that the gentamicin antibiotic is going to kill the S. epidermidis once we get this experiment underway. All right, so as far as supplies, what I'm going to use here is I have a very large petri dish here that's already been poured with the TSA, or tryptic soy agar. And as you can see, I've divided it into 3 portions here. So the PNC stands for penicillin, the NDB stands for novobicin antibiotic, and then the GNT stands for gentamicin. I have my S. epidermidis culture from the first lab that I pull it out of my fridge and it's been sitting for about an hour to come to room temperature. I have a pipette and some tweezers here sitting in alcohol sterilizing. I have this spreader thing that once I add some of the culture onto the petri dish, the agar dish, I'll use this portion to spread the culture around. Tea light to sterilize my tube. This ruler is going to be used later to measure. And then I have my antibiotic disc. So gentamicin, novobicin, and penicillin. So this Kirby Bauer diffusion test. So what I'm going to do is I'm going to place 8 drops of the epidermidis on the agar here. And then once I do that and spread it with that L-shaped thing, the dish will sit for an hour or 2, just so I can make sure that the culture absorbs well into the agar. And then in each of these little, I guess, pie-shaped things, or what you want to call them, I'm going to place the appropriate disc. So in the NVB, the novobicin disc is going to go, and the GNT, the gentamicin, and the PNC, the penicillin. And then the petri dish itself will sit for a minimum of 12 hours, up to 24, just to see kind of what's happening. What we're looking to see here is, well, I guess, what's described as a lawn effect. So since we're looking to see how effective or not effective the antibiotics are against the epidermidis microbe, what we're looking to see here is I'll try to place the disc, the antibiotic disc, in the center of each little section here. And assuming that the antibiotic is resistant or can kill or has the bacteriostatic effect, we'll see kind of like in that lawn effect that closest to the antibiotic disc, you'll kind of see, you won't see much growth of the microbe, but as we go further and further and further out, we'll see growth. So kind of think of this in a way kind of as like how a nuclear bomb works, for lack of a better explanation. But ground zero, or where a nuclear bomb hits, that's where the most destruction, the most barren an area gets. And as you get further and further and further out, there's still some destruction, but you have more and more buildings left, plants left, people are left alive. Same concept here. So the area closest to the antibiotic disc can kind of be considered ground zero, and there should be, if the antibiotic is effective, no microbe growth, bacteria growth, and then we'll go further and further and further out, and we should see some microbe growth. So now going back to how the antibiotics work. So let's go back to gentamicin. So gentamicin, it says it works against gram-positive staphylococcus. So since we know that epidermidis is gram-positive and it's the genus Staphylococcus, I expect to see some resistance or some impediments of growth. How the bacteriostatic effect of the gentamicin should work. Same thing with the novobicin, since it works against gram-positive bacteria, which epidermidis is. So, but we'll see. I mean, it's not 100%, but I expect it to work. The penicillin, maybe not, since it attacks primarily gram-positive bacteria. But how bacteria goes, how anything goes, the bacteria strain itself can mutate. Now, even with the gentamicin, I mean, there's also the thing that it affects gram-negative bacteria, but it also affects gram-positive staphylococcus. So we'll see. I mean, I'm hopefully confident that at least we'll see some of that lawn effect with the gentamicin. All right, so that being said, I'm going to flip the camera angle, put on my PPE, and then we're going to go ahead and get started with the experiment. All right, so the first step of this process, as I said earlier, is to add the epidermidis culture onto my agar in the petri dish here. So I have my pipette here that's been sterilizing for a good at least 5-10 minutes. So I'm going to expel all of the alcohol here And then just kind of bang it dry. Okay, kind of set that to the side for a minute. So, Petri dish, epidermidis culture. I'm going to take the lid off the dish, and then I'm going to flame the tip of this test tube to sterilize, as I've done before. Okay, so that part's done. Now I'm going to draw up about 5 milliliters of the culture solution. Okay. All right, and what I'm looking for here is 8 drops into the center of the dish. So 1, 2, 3, 4, 5, 6, 7, and 8. Okay, the rest is going to go back in. And then this is going to go into a bucket of bleach. I'm going to sterilize this again. Okay, lid on and set it aside. Now, I'm going to grab my little L-shaped shredder thingy out of here. And I'm going to use the L portion to spread the culture all over the Petri dish. All over the agar, just to make sure that every bit of it is exposed, so to speak. Okay, this is now going into bleach, and my lid's going back on. All right, now that Petri dish is going to sit for an hour or two while I so to allow the Staphylococcus epidermidis or the epidermidis to completely soak in, and then I will come back and apply the antibiotic discs. All right, so my Petri dish with agar has sat for 2 hours. So by now, the S. epidermidis, Staphylococcus epidermidis culture from my test tube has thoroughly soaked into the agar. So the next step is I'm going to add in each of the antibiotic discs. So I have my tweezers here that have also been sitting for almost 3 hours. Just sterilizing in alcohol that I'm just going to remove and tap dry here against the cup. Okay, so I'm going to start with my penicillin disc, which is here. Just gonna kind of gently move it to the front of the top of the bag so it's easier for me to get out with the tweezers. All right, so I'm gonna take the lid off the dish here, and I'm gonna do my best to put it in the center of the P and C region. So lid off. bag's now open, and then using my tweezers, I'm gonna so that's my penicillin. Just gonna put this as best I can into the center. Lid on. These are going in for 30 seconds back in alcohol to sterilize. And then this bag can go in the trash now. So we're gonna wait for 30 seconds and then move on to my Novobiocin. And while we're waiting, I'm just gonna do the same thing here. Put my disc towards the top of the baggie here. Okay. So there's the disc now. And again, that's Novobiocin. Alright, so it's been about 30 seconds. I'm gonna take out the alcohol, the tweezers from the alcohol, bang them dry, bang them against the cup to shake off any excess alcohol. I'm going to do the same thing I did with the penicillin. I'm going to take the lid off the dish, open my Novobiocin bag, and then using the tweezers, Novobiocin disc, oops. And I'm going to put it as best I can in the center of the agar. That was the center. Lid on. This is going back in the alcohol for 30 seconds. Trash. And then the final one will be my gentamicin. And it's very close to the top of the bag there, just to make it easier for me. Okay, it's been about 30 seconds. So again, the tweezers are going out of the alcohol. Bang them dry. I'm going to get some alcohol on this glove, so I'm just going to change it. Okay, and then the same procedure as I did with the other two antibiotics. Antibiotic discs is the lid's going to go on. Going to open the bag of the gentamicin disc. Okay. Don't know why, but this is being a little more difficult than the other ones to grab. Okay, so gentamicin. Okay, and again, this is going to go onto, as best I can, the center of the area. Okay. All right, so that's done. All three areas have an antibiotic discs in them. This is going to go in a bucket of bleach. This is going in the trash. Now this petri dish is going to sit for 10 minutes, just to allow for the antibiotic discs themselves to really well adhere to the agar with the Staphylococcus epidermis. Then they will go into my incubation location, which is my dark, temperature-controlled closet of my home office for at least 12 hours, up to 24. And what I'm looking for here, again, is that lawn effect. It should be kind of like a white film around the antibiotic discs, either showing resistance or not, depending on the antibiotic and the effects of it against the gram-positive Staphylococcus epidermidis. So we'll reconvene in 12 to 24 hours. Good afternoon. My name is Ricardo Sandoval, and this is a continuation of the experiment video for lab number 10, antibiotic sensitivity Kirby Bauer diffusion test. So this is part 2 of this lab, and this part has nothing to do with with an actual experiment, so to speak. In this portion of the lab, we're talking about the fungus, which can turn into an antibiotic, that's called penicillium. So penicillium is the fungus, penicillium is the fungus which becomes penicillin, methicillin, other cillins that I can't think of at the moment, but penicillium or penicillin is kind of like the mother antibiotic, and then there are many other antibiotics that are made from it. Penicillin or penicillium started from mold on an orange. So in this portion of the lab, like I said, there's no real experimentation. Basically, it all has to do with this slide that says penicillium with Candida. So Candida, they are asexually produced spores. So spores, you can think of kind of like mold. So what asexual refers to, so we have sexual and asexual. So we know that when the letter A is before any word, it means without in medical terminology. So we know, for example, when humans reproduce, there's sex or intercourse involved, that the sperm goes into the ovary, and then cells begin dividing until a human is born 40 weeks later. In asexual reproduction, there is no combination of what are called gametes. So in males, the gamete is the sperm. In females, the gamete is the ovary. So sperm goes to ovary. So in asexual reproduction, that doesn't happen. So there is no change in species, no getting something from the father cell and the mother cell, so to speak. And the number of chromosomes from the original cell don't change. So the purpose of this portion of the lab is to do some research, basically, on how penicillium works. So basically the environment that it grows in, how it works, bacteria targeted by penicillin, economic value, how valuable it is in society, and then some pictures itself of the slide identifying Canidia and something called Phyalidae. And that's pretty much the gist of this portion of the lab. So this is kind of a picture of the Canidia and the Phyalidae. So since there really isn't much, there is no experiment portion and no results portion, this will be it as far as the video is concerned. for this portion of the lab. There will be a results video for the previous portion in which I did grow S. epidermidis on a 9-inch petri dish and exposed it to gentamicin, novobiocin, and penicillin. Three different narrow spectrum antibiotics. And then we'll go from there. Thank you.